what happens is that this glucose, this is a major carbon source, right? So this glucose is going to give two molecules of pyruvate, this one and this one, right? It is going to give two molecules of pyruvate. This particular molecule of pyruvate is going to form acetyl-CoA, and the other pyruvate is going to give oxaloacetate. Now when these two, acetyl coenzyme A and oxaloacetate, together will form the citrate in the Krebs cycle, right? So this is how we get, this is a very, very fundamental, very, what you call is, I'm not going, I haven't given the complete biosynthetic pathway starting from the glycolysis to the Krebs cycle. I have just summarized it in a very small step, like glucose is the carbon source. Then glucose is going to give two molecules of pyruvate. One pyruvate is going to form acetyl-CoA, another pyruvate is going to form oxaloacetate. And together this acetyl-CoA and oxaloacetate are going to form the citrate in the Krebs cycle. Now if we talk about the enzymes in the production of citric acid, this citrate synthase is a very important enzyme, right? It is the activity when there is a production of citric acid, then the activity of the citrate synthase, it increases, almost 10-fold increase आ जाता है जब ये जो आपका citric acid production होती है. क्यों? क्योंकि this is one of the key enzymes. Another key enzyme is pyruvate carboxylate, which actually converts pyruvate to oxaloacetate. So this is considered to be this enzyme is considered to be a key enzyme in the citric acid production. So these two enzymes are very important, right? So you should know that whenever there is an increase in the citric acid production, there is a 10-fold increase in the citrate synthesis production. Similarly, this pyruvate carboxylase is again the key enzyme, because this is an enzyme which is going to convert pyruvate into oxaloacetate, and oxaloacetate is very important for the production of the citric acid, right? So now I'm going to start with the fermentation. What are the process of fermentations? What are the different types of fermentations? What are the disadvantages and advantages of these fermentation for the production of citric acid, right? So before starting the description about the different fermentation processes, I would like to mention here that there are a few factors which actually affect the fermentation and your citric acid yield, right? So for the very first is trace elements. So these are the trace elements like iron, copper, zinc ions, manganese, magnesium, cobalt, right? So these are some trace elements which are actually very essential for the growth of Aspergillus niger, right? So if they're present, very nice, very good for the growth of Aspergillus niger. And also in this ferric iron, this zinc and manganese, they support the production of the citric acid. They actually enhance the citric acid production, right? Then when we talk about the pH should be less than 2.5 for the production of citric acid. If you want a good production of citric acid, good amount of citric acid, then pH has to be less than 2.5, right? So if the pH is 2.5, there are two benefits. One thing, the production of citric acid is increased, it is enhanced. Another thing, that below 2.5 there are very few organisms which are actually able to survive. So the chances of contamination are very, very low in this case, right? So this is something which is advantageous to our process, right? Because we do not want contamination in our fermentation, right? So if the pH is 2.5, then the production of byproducts is also checked. But when the pH reaches to 4, or say it increases more than 4, then what happens is, then there is a production of oxalic acid. Then there is a production of oxalic acid. Then after this, if the pH gets increased to 6, then there is a production of gluconic acid. Right? Then the gluconic acid starts accumulating at the expense of the citric acid. Citric acid will now not being produced. At the expense of citric acid, gluconic acid will be produced at pH 6 or more than 6. Then at the expense of citric acid, if the pH is 4, oxalic acid will be start accumulating. But with pH 2.5 or less than that, there will be good production of citric acid, right? This is about the pH, which is a very, very important factor. This is often being asked in exams also, that what is the right pH, which is the optimum pH at which the production of citric acid is being commenced, right? Then we talk about the dissolved oxygen. Now, oxygen, sparging is very, very important. more the dissolved oxygen tension, more the production of the citric acid, right? So it is directly proportional. The amount of pure oxygen being sparged into the fermenter, जितना fermenter में आप pure oxygen दोगे, जितना ज़्यादा dissolved oxygen का tension बढ़ेगा, उतना ज़्यादा आपका citric acid production जो है वो increase हो जाएगा. So it is directly proportional, right? Then when we talk about the nitrogen sources, so we have, we have ammonium salts. We have ammonium, no, this will increase. We have ammonium salts, then we have urea, then we have nitrates. All these are very, very good sources of citric acid, and then they support the citric acid production nicely. Yes, there is one more point, that in case of molasses, if you are using molasses as a carbohydrate source, then there is no need to add, separately nitrogen source. You don't need to add them separately. Okay.